We're live.
Hello, good afternoon and welcome everybody to our virtual RCSI campus for the afternoon. Welcome to this, our third event in RCSI's Women's Network, Ensuring Women's Voices Are Heard series, an initiative of our Equality, Diversity and Inclusion unit. My name is Louise Keating. I'm a lecturer in the School of Physiotherapy and a clinical physiotherapist, and the Women's Network co-chair alongside Dr. Jamie O'Sullivan, who you'll meet a little later. The format for our 90 minutes to come is 3 blocks of a panel of 3 to 4 presenters, and each block will follow with a Q&A. So we'd really like for these research stories, hopefully, to ask you to interrogate the researchers with your questions, because there's lots to share with you this afternoon. So please feel free to populate your questions into the chat during this session. Alternatively, you can also use our Twitter handle, hashtag RCSI Discover. And for those of you who are listening back to this after the event on YouTube, you're welcome to this event also. So our first block, we're going to focus on the theme that involves our health across the lifespan. And we'll start with 4 researchers who will take us on their journey in how they have tackled health problems from infancy to older adulthood. And so we'll start with Dr. Jennifer Dowling. Jennifer is a lecturer in immunology and biotherapeutics in our School of Pharmacy and Biomolecular Sciences, and she's going to share with us inflammation, why it's big in her life. Over to you, Jennifer.
Thanks, Louise. Hi, everyone. As Louise mentioned, I'm a lecturer here in RCSI, and I also conduct research on our immune systems and how the process of inflammation can really be studied so that we can understand and treat disease. And it's hard to imagine actually that when I was growing up, I never thought about becoming a scientist. I always knew I wanted to travel, but I didn't really know exactly what I wanted to do in college. So I ended up enrolling in a course of international languages and marketing. And so that made sense to me because international and languages, I get to travel. But unfortunately for me, it just didn't work. The grammar, I couldn't get to grips with the grammar, and I wasn't really enjoying it, to be honest, if I'm honest. And what happened was then I took a bit of a time out and I said to myself, right, if you had to go to college every day and listen to somebody talk about something, what would you like to listen to and what would keep you engaged? And so what happened was I had done physics for the Leaving Cert, but I'd never done biology. And I was always interested in how, things work and watching the Discovery Channel and things like that. So took a bit of a time out, spoke to friends, family, people I knew in college, a couple of members of staff in DCU, and I enrolled then in a degree in biotechnology. And to be honest, I was hooked. I never looked back. By year 3, I was actually sent over to Philadelphia to work, and so that got me to travel straight away. I hadn't even finished my degree, and came back and decided I have to do a PhD in research. I just love it, and so I had to decide then, what did I want to work on? And I knew I didn't want to work on one protein or one organ. I wanted to work on something that really impacted the whole body. And for me, we were learning about the immune system, and the immune system was really it was that exciting to me that it impacts your whole body. So I did a degree, my PhD in immunology at DCU, and I focused on a cell type known as a macrophage cell. This guy here I'll talk about later. And these guys are really important for the process of inflammation and controlling inflammation and mediating disease in the body. And so after the PhD, or actually during the PhD, I had a fantastic supervisor, Professor Christine Loscher, and I got to go all around the world. I presented in Brazil, France, Germany, went to Turkey, went to Greece. So, as it would happen, I got to travel and do what I love to do and to do something I love doing. Then I ended up in Australia for 4 years, where I really honed my skills over there on the signaling and mechanisms in this cell type and how they contribute to different diseases. And so now I'm back in RCSI, absolutely where I want to be, doing exactly what I want to do. And just to fill you in now a bit on that inflammation that I'm doing the research on. So these guys, these macrophage cells, are really, really interesting. So essentially, when you have an infection, for example, in your body, these guys come into the area. They get signals that tell them, right, you need to gobble up the infection, the bacteria, and you need to really promote inflammation as a process to clear the infection. And what's really nice is it's really a life cycle that these cells have. They go through this life cycle of inflammation where by the end of it, they end up in this kind of steady state where they calm down the infection and resolve inflammation in the body to get things back to normal. And it's really my research really focuses on how do we keep these cells in this state, because we know, for example, in autoimmune diseases such as arthritis, there's a constant signal to keep the cells in this red state, and that causes low chronic grade inflammation that is really detrimental for patients. So if we can understand the mechanisms to keep these macrophage in the green kind of healthy resolving status, we can promote and repair disease. And what's really cool for me is that in the last couple of years, people have identified that this cell type can actually promote repair not only in tissues in the periphery of the body, but they can actually promote repair in the brain. And I think that's really cool. So my latest research is working to understand how, what is the role of these cells in the immune system in the brain during cases of inflammation. And one particular project in the lab is looking at the role of these cell type and the immune system during seizure. And so in seizure, you have low-grade, sometimes chronic, low-grade inflammation. We know this. For example, when babies do not get enough oxygen, sometimes after birth, they can have seizure. And certainly people with epilepsy have seizures. So one of the projects is looking at that. And just to show you some really cool data from the lab, I'm just going to share something with you here. Our latest research, bear with me there. Our latest research has identified a really cool protein in those green cells that helps them, basically a protein in the cells of the immune system that helps them stay in that status. So this is why inflammation is big in my life. Essentially, this is an example of a Western blot, and it's a really cool technique in the lab where we can profile a patient's proteins. And what I want to show you here is that in healthy patients with epilepsy, you can see very high levels with the black bands of this protein that keeps the cells in the green status. However, when we look at patients that have epilepsy, the levels of that protein are actually really low. So my research is working to develop therapeutics that we can possibly bump that protein back up to a normal level and help the inflammation and possibly the onset of seizures in people with epilepsy, or indeed babies that have epilepsy sometimes after birth. And so just to finish up, to say that that's why inflammation has been big in my life, and I've been very lucky that it's allowed me to travel all over the world. Little did I realize I've made exciting discoveries, and as I said, it's big in my life, and I believe it will continue to be big in my life, as I'm just launching my own research group here in the college, which I'm calling the Brain Inflammation Research Group, or Biggie for Brain Inflammation Group Ireland. I have launched a couple of socials for the lab, and you're more than welcome to follow me on my personal Twitter account, and I've launched a couple of sites for the lab. And I hope you enjoyed that synopsis of my research, and I'm happy to take any questions at the end of the session. Thank you.
Brilliant. Thank you, Jennifer, for getting us started this afternoon. That big is definitely going to stay in my brain now. So thank you for that. It was really interesting. Great. So please feel free to post your questions in the chat for Jennifer before we hear from Niamh Arthurs. So Niamh is a registered dietitian and researcher across the School of Physiotherapy and the Division of Population Health Sciences here in RCSI. And she's going to share her research story. A childhood obesity researcher. Now, how did that happen? Niamh, over to you.
Thanks, Louise. And when I was young, people often ask me, what do you want to do when you grow up, or what do you want to be? What do you want to do after secondary school? And when I was young, my answer very confidently was that I wanted to be a warrior princess. I'm just trying to share my screen here. Hopefully it's sharing now. A warrior princess like Xena, pictured here in this photo. And people have stopped asking me, what do I want to do, or what do I want to be when I'm grown up? So I guess that's a clear indicator that maybe this stage I'm grown up. But when I got to secondary school, I started to think, you know, just in case the warrior princess role doesn't work out, it'd be good to have a backup option. And I've always really loved animals. This is Pugsy, my rescue dog, and Pugsy has a brilliant personality. In the first image here, Pugsy had just had a lovely bath, he was clean, and then he decided that he didn't want to be clean, so he went outside, rolled in a lot of muck and dirtied himself. In the last picture there, Pugsy, you can see him pressed up against a radiator, and because he realised that he could warm himself in this way. So Pugsy taught me how he can be rebellious, but also cute and clever. I ended up studying nutrition and dietetics. And if you'd said to me when I was doing my degree that I would end up as a researcher, I would have been on the next flight to Canada or New Zealand. But I am a firm believer in what's meant for you won't pass you, or, you know, that life works in mysterious ways. And in my work, I came across obesity a lot. And like many people, I thought that the treatment for obesity was just to eat less and move more. But clearly, we still have the issue. It's a global epidemic, and it's not being solved by eating less and moving more. I came across the Association for the Study of Obesity on the Island of Ireland, or the ASOI, and I began to learn about all the different contributing factors and complications of obesity, and that there's so much to know about it. I became a committee member of the ASOI, and I also learned the importance of people first language. And what I mean by that is that when we're describing people, we might say a smart person, a funny person, a tall person, but we wouldn't say a heart diseased person or a cancerous person. And in the same way, it's not respectful to say an obese person. And the preferred language by those with obesity is actually to say a person with obesity or people living with obesity. And that's just something to be really mindful of, because language really matters. An opportunity came up for what would be for me an absolute dream role as a childhood obesity researcher, having gained so much knowledge from being an ASOI committee member. I spent weeks preparing for this interview because it was also a fantastic opportunity to work with an absolutely phenomenal team. And my passion and enthusiasm must have shown because I got the role. And this led me to work on a Sláintecare childhood obesity project in which we developed online childhood obesity training, which is free and CPD accredited for healthcare professionals in Ireland. And you can access this free online and CPD accredited training on our website, childhoodobesity.ie. And there's also some really nice resources on our website too. Everyone still see my screen?
No, but we can hear you, Niamh. We are seeing Austyn's screen a little ahead of time, so but we can hear you fine. Okay,
I'll just continue. I'm not sure why that happened. I'll try sharing again and I'll just continue anyway. Okay, trying to still share my screen. Hopefully you can see it. Anyway, the Sláintecare work led me on to my current role on the Landscape project. And what we're trying to do in the Landscape project is identify enablers and barriers for the provision of obesity treatment services for children and teenagers in Ireland. And we also want to try and seek insight from healthcare professionals and health managers who currently work with children and teenagers in a healthcare capacity. And then we want to identify the supports that might be needed or are already available to enable this provision of childhood obesity treatment services. So if you are a health professional or a health manager and you think you might be able to help us, I would really encourage you to go onto the childhoodobesity.ie website. And if you look at the news section, you'll be able to access information on the Landscape project. I don't work because when you love what you do, you won't work another day in your life. And part of the love for my role is that it's in an area that I'm really committed and dedicated to, and I really want to contribute and bring about changes to improve the lived experience by those with obesity and by families affected by obesity. But also, it's down to the team, the people that I get to work with. And I can't emphasize enough how important working relations are. I often refer to my colleagues as the dream team or the A-team. And yeah, another thing that I just want to mention and extend my gratitude to is my family. My parents, my siblings, my sisters-in-law, my brothers-in-law, my extraordinary nieces and nephews, and all of the adorable animals over the years. Because I wouldn't be who I am or where I am today without them. And in the words of the Take That song, never forget where you're coming from. I'm Niamh Arthurs. I'm a registered dietitian and I'm a researcher. Thanks for your attention. I'm happy to take any questions.
Brilliant. Thank you so much, Niamh. That was really fantastic. Brilliant. So listen, we will keep inviting the audience to share their questions for the panel, and do so now while you're listening to their presentations and prompted by what they're saying, and we'll hold those questions until we get to our Q&A shortly. So next we'll invite Dr. Austyn Matheson, who is a postdoc researcher in the Tissue Engineering Research Group here in RCSI, to share with us her story, how she's building bone and constructing cartilage. Over to you, Austyn.
Hello. Hello, yes, my name is Austyn Matheson, and I am a postdoctoral researcher with the Tissue Engineering Research Group at RCSI, and I study biomaterials to build new bone and construct new cartilage in our bodies. So if you know a bit about me, I'm constantly moving. I'm from Canada, and so far I've lived in 5 different cities. I move around a lot. I'm always walking, running, or biking on my way to work, to research, to fun, or the next adventure. I really only have 2 speeds, fast and faster. In all, the ability to move, my mobility, is incredibly important to me. So, the virtual audience, what about you? When you think about your day-to-day, what are your favorite things to do? What kind of movement brings you joy and excitement? So just for a moment, I want you to imagine that you were no longer able to participate in those activities, that moving from point A to point B was very challenging. Well, joint disease or osteoarthritis will affect an estimated 1 in 4 adults in Ireland by 2030. So evidently, this will have a significant impact on the quality of life of those that are affected. And this is why I'm so excited about my research here at TURG with RCSI. We are making biomaterials that will help prompt the regrowth of diseased bone or cartilage in the body so that we can repair the mobility that is lost to various conditions. The biomaterials or scaffolds that we make are predominantly made of collagen. So collagen is the building block protein of the body. It provides the structure to house all the different tissues and organs and our cells. So to make these scaffolds, we blend the material and freeze dry it to make these perfect porous sponges. These are implants that can be placed into the empty defects of cartilage or bone that's missing, and they provide a framework so that cells can enter and complete regeneration can be made. So once the tissue is regenerated, it's no longer distinguishable from the surrounding implant. So it's basically the scaffold disappears. We make each biomaterial specific to the body part we hope to regrow. For bone, we add minerals like hydroxyapatite that increase the stiffness, and for cartilage, we add biomolecules like hyaluronic acid that help the stem cells that reach the biomaterials become cartilage cells or chondrocytes. And within TURG, we have even made a 3D printed biomaterial that has been implanted in rats, sheep, horses, and even humans with clinical success. So, like all technologies, there's still room for improvement, and that brings me to my research here at RCSI. This work started with the collaboration during my PhD. I was studying a protein called lubricin. So this is a molecule that's located on the surface of cartilage and in synovial fluid. It helps lubricate the joint. So lubricin lubricates. Okay. So during this collaboration, I worked with a team where we demonstrated that adding this molecule lubricin to the TERG scaffolds could significantly reduce friction. So it made the scaffolds more slippery. So this was a great finding. However, there are some limitations. This molecule is very expensive to make, and we can't add enough for a therapeutic effect that's long lasting. So now that brings me to my current research. My upcoming research as part of, I'm an Admire Mary Curie Sklodowska Fellow with the Amber Center in RCSI. So I'm going to be working to design a plasmid DNA to deliver lubricin plasmid. So what is a plasmid? A plasmid is a circular piece of DNA that contains the specific instructions for cell synthesis. So first, we encapsulate or combine our DNA with nanoparticles. These are called vectors. These vectors help protect the DNA so that it can be transported into the cell and enter the nucleus. These particles are so small, they're nano-sized in fact. So this is why they're called nano. So after making them, they're added to the TERG scaffolds. So now a few complex images. These nano complexes work by entering the cell cytoplasm and then into the nucleus, which is the control center of the cell where the protein is transcribed and translated. So in basic terms, our overall goal is to instruct the cell to make its own lubricin. This means that we no longer have to add it onto our scaffolds. So overall, my goal in this research is to develop a gene-activated biomaterial. So for the first time to deliver a lubricin plasmid by non-viral nanoparticles in order to regenerate cartilage tissue that has superior frictional properties. So thank you very much.
Fantastic. Thank you, Austyn. Just time for our fourth speaker for this panel before we move to your questions. We're going to now hear from Natalie McEvoy. Natalie is a PhD scholar at the School of Nursing and Midwifery and also a clinical research nurse in anaesthesia and critical care. And she's going to share with us her story from nurse to nurse researcher. Over to you, Natalie.
Good afternoon, everybody. It's lovely to be here today to talk to you all. So I'm just going to share my screen with you now. All going well. And that's perfect. Hopefully you can see that now. So thank you for the introduction, Louise. My name is Natalie, as you've just said, and I am conducting my PhD in School of Nursing and Midwifery in RCSI, and I'm also working on some research in the Department of Critical Care and Anaesthesia. So I'm just going to share my story with you for the next 5 minutes or so. So, sorry now, I have a delay in my slides. So I'll be giving a brief background to sort of my career and, you know, kind of where I started out and where I am now. I'll be talking about why I chose a career in research, and I'll also be talking about some COVID-19 related research. It's obviously quite topical at the moment, and I'll be talking about one project in particular. So to start then, I suppose, I studied general nursing in DCU, and I graduated from that in 2015. And after that, then I started working as a staff nurse on a neurology ward. So I really loved working there, and I completed my postgraduate diploma and my master's in neuroscience nursing then. But I moved to the ICU in 2016, a neuro ICU, and I absolutely loved it there. I loved everything about it, the team, the work, just everything. And then I started working as a clinical research nurse in critical care in 2018, and then I started my PhD in 2019. So why research then, or why did I pursue this? Because it's not the most common career pathway for nurses. Many nurses they wouldn't consider a career in research, because there's so many opportunities in the clinical world. But for me, I always found that I was asking these questions: who, what, when, where, why, and how. Particularly as a junior staff nurse, I would ask people more senior to me, you know, well, why do we treat patients in this way as opposed to this way? And the answer was always, well, That's what the evidence says we should do, and that's what the research says we should do. And, you know, people make reference to evidence-based practice. And for me, I was confused. I thought, where is this evidence coming from? You know, who generates this evidence? And I just really wanted to know more. So that's why I wanted to get into research then, was to try and answer some of these important clinical questions and to be involved in generating some evidence. So clinical research has so many benefits. For me, it is being able to still maintain patient contact, which is really important, and I really, really enjoy that about my job. And there's exposure to the clinical environment, which is constantly changing. So if you remove yourself from the clinical environment, even for a number of weeks, by the time you go back, it's so many things have changed as a result of updated evidence and research, I suppose. And we're constantly trying to improve the care that we provide to our patients. So answering important clinical research questions, as I've just mentioned, and then witnessing the findings of research being implemented in real time. And I suppose the COVID-19 pandemic has been the perfect example of that, where evidence is generated and we have, you know, improvements in how we can care for our patients, and immediately then that care is implemented into the real life clinical world. So it's really amazing to see and to be a part of, and that's why I love my job. So I've been involved, along with my PhD, I've been involved in some COVID-19 related projects over the last year or so as well. And I know inflammation is it's a topic of discussion today, but one of the things we did last year was we tried to look at the inflammatory response to severe COVID-19, among other things like prone positioning, oxygenation, and we also looked at skin injury in healthcare staff wearing PPE or personal protective equipment as well. So I'll very, very briefly chat about one project in particular. There were a few, but I've chosen this one because they were just nearing the end of it now. So it's very, very near being published, and we're close to finding out our results. And it's kind of a mouthful when you read the title, but basically this was the first approved clinical trial in Ireland to test a therapy for COVID-19. And it involves patients with acute respiratory distress syndrome, or ARDS, which is quite difficult to explain in simple terms. But basically, a lot of us would have seen in the media and on the news, people, you know, images of people in the intensive care really, really sick with COVID-19, and many of these patients will have had ARDS. And basically, it is when the lungs, there's a buildup of fluid, and then therefore the oxygen can't get into the bloodstream and to our tissues and our organs that rely on oxygen in order to function and to keep us alive. This trial involves the administration of a drug, and in this case it is called alpha-1 antitrypsin. So this is a protein. We actually produce it naturally in the body, in the liver, but it has anti-inflammatory properties. So these patients with ARDS, there's a lot of inflammation going on. So the idea behind this trial is that by giving this anti-inflammatory medication, we may, you know, it may help with that inflammatory process that's going on. Very, very quickly, so it involves randomization. The most simple way to describe randomization is like tossing a coin. So some patients will get a placebo, like salty water, also known as saline. So some patients will receive saline and some will receive the active medication. And it is, of course, it's double blinded, which is the gold standard in terms of clinical research and randomized control trials. It means the researchers and the patients do not know whether or not the patient is receiving the active agent or the placebo, in this case it is saline. So the benefits to that are that it reduces bias in the research, and hopefully we can produce really sound, good quality results that we hope to share with people in the coming weeks. Thank you very much for listening to my research story. I've left my Twitter handle there and my email address, and I also look forward to taking any questions at the end.
Fantastic. Thank you so much, Natalie. And actually, we might start with you while your mic is still on. ICU is obviously a really challenging environment to work in, none more so obviously than the last 18 months. But how do you balance the challenge in researching within ICU? It must be a really challenging research environment as well.
It is. It's quite tough, and I suppose it's a really it's a very intense environment. They don't call it intensive care for nothing, I suppose. So it is a real challenge. It's quite stressful. There's always so much going on, but doing research it's almost, especially since like the beginning of the pandemic, research has just become, the nurses will often say to me, like, what research are we doing now? It's just become part of standard care almost, which is really incredible to see. Because before the pandemic, I sort of felt as though, research was a bonus in the clinical setting. Like if we did participate in some research studies, it was fantastic. But if we didn't, it was okay. Whereas now we need research. Like with COVID-19, we've had to learn so much as we go on, and like the care we'd say that is provided to patients now in the ICU is quite different to what would have happened in March of last year. Many of us will know that steroids are now something that is routinely given to patients with COVID-19 as a result of a major clinical trial that happened and was published, I think, in September or October of last year. So it's a challenging environment, but I do think that it's been just accepted as something, and it's been embraced, the research culture, which is fantastic to see.
Great, thanks, Natalie. Okay, the question has come in for Jennifer. Jennifer, how do you think you'll be able to alter the expression of a protein? The protein that you showed was lower in the epilepsy patients. So over to you, Jennifer.
Yeah, no, that's an excellent question, Louise. So what's really interesting about that protein is that we know in environments of inflammation, the DNA of that protein is actually targeted in specific ways to be degraded. So then we don't get any protein translated. So what we're developing at the moment is what we call basically a DNA blocker targeted to the sequence of that protein so that it's protected when there's inflammation, so that you'll still have levels of the protein in the system, hopefully. So that's what we're endeavoring to do.
Fantastic. You make it sound very easy, but it's very complex. Thank you for that. Niamh, I'm going to come to you next. Really, the role of a researcher and the work of a researcher spanning so many years obviously is filled with challenges along the way. I suppose for anybody listening who's interested and perhaps drawn to a career in whether it be healthcare and research, as Natalie suggested, or beyond, specifically purely in research, what has been your biggest challenge as a researcher, I suppose, to overcome to date? Because that's a really useful insight for people to hear about.
Yeah, thank you for that question, Louise. I suppose probably one of the biggest things is learning that I found that the more I started to learn in a researcher role, the more I realized what I don't know. And there is this thing called the Dunning-Kruger effect, or the Dunning-Kruger effect. And it is that the more we learn, the more we realize what we don't know, and then that can sometimes reduce maybe our confidence. But I would say accepting that it's okay not to know everything, and that is why, again, I get to work with fantastic, fascinating people that I know I can turn to and ask for support or for guidance, and they're always so helpful and willing to give that. So being okay with not knowing everything, but being open and willing to learn. And once I accepted that that's okay, I don't have to know everything, and that I can learn as I go along, I'd say that was one of the greatest things that I found challenging.
Fantastic. Thank you, Niamh. And then finally, to the last member of our panel, Austyn, really conscious of how, as physios, for example, we're seeing people straight after perhaps having an injury to cartilage, or maybe even later on when their articular cartilage isn't working so well, or maybe even post-surgery for rehab and things like that. So I suppose where you're at this stage in terms of tissue engineering research, for you, I suppose I'm interested, what's the holy grail for what you think is possible down the road when it comes to optimizing our joints for our lifespan?
That's a big question, and I have to say I'm not entirely sure. The work we work on, so joint replacements are excellent at doing their job, restoring mobility and allowing people to return to walking or normal activities again. So the work we're doing with TURG on our biomaterials, we're working on them with the hopes that they could help regenerate tissue in patients that might be younger before they need to go get a full joint replacements. So it's hopefully a therapy that can be that intermediate step before they need or to kind of replace it. So that's the work we're doing.
That's really exciting and a very optimistic note to end this first panel discussion on. So my thanks to our speakers. I'm going to now hand you over to Dr. Jamie O'Sullivan, who is co-chair of the Women's Network and a star research lecturer in the School of Biopharmacy and Biomolecular Sciences. Over to you, Jamie.
Thanks very much, Louise. Good afternoon to all of you joining us online. As Louise said, my name is Jamie O'Sullivan. I'm a researcher and lecturer here at RCSI, and I'd like to very much welcome you to our next session, which is going to be focused on human diseases. So in this part of our Discover RCSI event, we will hear from researchers who work on cancers, including blood cancer, the application of new technologies in cancer diagnosis, as well as gut health and inflammation. So if you have any questions, I really urge you to use the Q&A box to submit them, and we'll do our best to get to as many as possible after we hear from our three speakers. And if you're engaging on social media, please use our hashtag RCSI Discover. So without further ado, we'll move on to our first speaker. So this is Lindsey Flanagan, a PhD candidate from the Department of Physiology and Medical Physics. And today, Lyndsey is going to present on multiple myeloma, a bench to bedside story. So over to you, Lyndsey. Lyndsey, I think you may be muted.
There. You would think that all this time with using Microsoft Teams because of COVID, I would have gotten used to that by now. I'd just like to say thanks for the introduction. I'm delighted to be here, and I'm delighted to be taking part in the RCSI research stories. So today I'd just like to talk to you a little bit about the work that I do in multiple myeloma. So first off, multiple myeloma, it's a blood cancer, and it affects the cells that are responsible for producing antibodies that help fight off infections. And these cells are found within your bone marrow. And so in Ireland, we have 305 new cases per year with 169 deaths per year. So patients with multiple myeloma, they can experience a range of symptoms, which can include fatigue, renal failure, bone pain, and increased infections. And so multiple myeloma is an incurable, poor prognosis cancer with less than 50% of patients surviving 5 years after their initial diagnosis. So multiple myeloma cells, they are reliant on BCL2 proteins to help them avoid cell death. And the way that they can do this is by increasing the expression of BCL2 proteins to help them survive. And so a drug that I work with in my project is called ABT199. And this is a drug that can target cells that are reliant on BCL2 proteins for survival. So the aim of my project is to try identify a new combination of drugs to help ensure a longer treatment response in multiple myeloma patients. And today I'd like to talk to you about a really interesting patient sample that we had. So in collaboration with Professor Siobhan Glavey of Beaumont Hospital, we have ethical approval to receive patient bone marrow samples into the lab. And this patient, they had a 2-year history of multiple myeloma. They were resistant to standard therapies, and they'd actually developed a really rare and aggressive form of multiple myeloma called secondary plasma cell leukemia. And when they presented into the lab, they were experiencing this blurred vision. And this can happen when your blood becomes thicker than normal. So what happened was the multiple myeloma cancer cells, they can produce excess proteins, and this can cause the blood to become thicker than normal. And as a result, this patient was experiencing blurred vision. So when I got the patient bone marrow sample into the lab, I'm able to isolate the myeloma cancer cells from the bone marrow sample. And from there, we're able to treat the myeloma cells with a range of different drugs to see if there's any drugs that this patient sample will potentially respond to. We also have this technology in the lab called BH3 profiling, and what we were able to do with this sample is show that this sample was really dependent on these BCL2 proteins for survival. And what we found when we treated the sample with the drug ABT199 was that there was significant death in the myeloma cancer cells. So in this bench to bedside project, in one part in the lab, so the patient sample, we treated it with different drugs, and we were able to show that this patient had an incredible response to ABT199. And then at the same time in the clinic, Professor Glavy did analysis on the patient sample, and the patient was actually started on a regimen of ABT199, and they were given this for 10 weeks, and incredibly, this patient went into remission. So what we hope to achieve is to use the technology that we have in the lab, the BH3 profiling, to try identify myeloma patients that are dependent on BCL2 proteins for survival, and that will hopefully respond to ABT199 treatment so that we can make more survivors. And this project is generously funded by Breakthrough Cancer Research, and I've been delighted to be able to share a little bit about the research that I do, and thank you all for listening.
Thanks, Lyndsey, that was fabulous. So we will move on now to our next speaker, and next up is Camille Hurley, a PhD candidate at the School of Pharmacy and Biomolecular Sciences. And today, Camille will be presenting her story on life choices, artificial intelligence and breast cancer.
My name is Camille Hurley and I'm a final year PhD student here in RCSI. I'm also an avid gardener, I like to dance and I like to dress up. And the title of my talk today is, let me see here, a PhD in digital pathology and artificial intelligence in breast cancer. Who would have thought? Because I certainly didn't think so at all. So I'm going to talk to you a bit about how I got here today and a bit about my research. So I did my leaving cert in 2009. I did kind of a funny mix of option subjects. I only did biology as one science subject. I did 2 languages. I love languages, and I did ordinary level maths, which was also kind of unusual for people who go on to study science. And then I did 1 month of applied psychology in UCC, but that definitely wasn't for me. So I left that and I didn't really know what I was going to do. And it was actually my mom who suggested I go to Germany to go au pairing. So that's what I decided to do. And I planned to go for 6 months, but I was getting on really well. I was with a great family. I was in the middle of nowhere, but I really was enjoying it. And I decided to stay a bit longer. So I stayed another year and I got a waitressing job in the next village. And this was really a brilliant experience for me to learn German and just have a completely different, I guess, type of life for a year or so. But I did have to come back and I decided to go to UCD. I was going to do neuroscience because I was still kind of interested in brain side of things. But I ended up kind of veering off into the pharmacology route instead. So pharmacology is all about how different diseases and how drugs work in diseases. And I thought it was really, really interesting. And I did enjoy this, but it was pretty tough, especially in the final year. And while most of my friends were thinking of what masters or PhD they were going to do, I really knew I needed a break. And I was like, no way, am I definitely never doing a PhD. I just want to get out of here. So I'd had a waitressing job all the way through college, and I decided to work full time for 6 months waitressing to save money to go traveling in South America. And this was just definitely something I had to do, and it was absolutely the right decision for me. And then when I came back, I emailed my old lecturer from UCD for a reference because I knew I had to start looking for a job. And he actually told me he was in RCSI now, and he offered me a job as a research assistant. And I did really want to have another experience working in a lab that was, unrelated to my undergrad. So I started this job just to see how it would go. And then the opportunity to do the PhD came along. So like I said, I really didn't think I would ever do it. But when the opportunity presented itself, there was, one thing I have always known is that I want to do something with my career and with my time that's worthwhile and meaningful and beneficial to people. So of course, research is a part of that. And I also knew with the PhD there would be opportunity to move around again, to travel at conferences or work in different labs like Jen was talking about earlier. And indeed, I did get to live in Paris for about a year in total. I got to work in a lab there with our collaborators in the centre of Paris. So it was really, really great experience to work with another group. Again, I got to speak French, so I was practicing my languages again. Yeah, so it wasn't the easiest way to do the PhD, but I'm definitely glad I did it this way. And the reason I'm kind of showing this today is because I just want to kind of reinstate that there really is no one correct path to take. And while everyone might be doing, you know, going straight on to do things, if it's not right for you at that time, you can take a break or, you know, do what feels right for you. And this is kind of the ethos that I try to live by. And I'm a bit older than most people in my year now doing the PhD, but that doesn't bother me at all. And yeah, I'm really glad I did it this way. And don't get me wrong, I was really terrified before I did each of these things. It sounds like now that it was all really easy and great, but I was scared. But you know, I think when we do the things we're scared of, it's where the best growth comes from. So anyway, where am I now? I'm at the end of my PhD. I'm about to start writing my thesis. I'm going to talk to you a bit about my work. So what is pathology? So pathology is basically the study of disease. In my case, it's the study of tumours. And what a pathologist does is they can, we take a piece of tumour, like this one here, I hope you can see it, and we can look at the tumours down the microscope. And the digital pathology is basically when we can take these pieces of tumours and we put them in special scanners, and then we get an image of the tumour on the screen. And this is really great because we can move around the screen, we can look at different parts of the tumour, we can look at them with our collaborators and with other scientists. And why we're looking at images is because actually images of tumours give us loads of information about the disease, and we can find different patterns and different characteristics of the tumour when we're looking at these images. And a lot of pathology now is using artificial intelligence. So this means different AI softwares that are being used to find things in the images that maybe wouldn't be recognised by the human eye. In my case, I'm using artificial intelligence to count thousands and thousands of immune cells in my tumour images. So my research question is basically which type of immune cells are entering breast tumours and what effect does this have on patient outcome? So to give you an idea of what this looks like, this is a piece of a breast tumour basically that was taken from a patient at a time of surgery. And if we zoom in on one zone, we can see here with the different colours, different types of immune cells. And I must say that the different colours are not naturally occurring. The colours are on the cells as a result of an experiment that I did in the lab on the tumour before I scanned it for the image. And then I'm using artificial intelligence to help me count these immune cells, as I mentioned. So here basically is the AI software, as we can see by these computerized little circles, counting these different types of immune cells. So I spend a lot of my time, or I have spent a lot of my time actually training the AI software and teaching it how to recognize the different cell types. And this is then an image of it performing well and counting the different types. So I must say thanks, of course, very much to everyone who contributes and supports to this work. Of course, no one works in isolation. Definite shout out to my family as well, inspired by Niamh. Thank you to them for putting up with me during these 4 years, and thank you all very much for listening.
Thanks, Camille. That was really insightful. Thanks for sharing your journey with us, and I'm sure that's probably directly applicable to a lot of people who are taking the next step in their careers in college and whatnot this coming month. So we will move on to our next speaker, and I'm delighted to present Jessica Smith, a PhD candidate who is going to present her research on chronic diarrheal disease, food for thought. So Jessica, over to you.
Thank you. Can everyone see my slides?
Not yet. I think they're about to be put on screen. There we go.
Perfect. So hi everybody, my name is Jessica Smith and I'm a final year PhD student here in the Royal College of Surgeons. My journey into research really started about 7 years ago when I was in the 4th year of my undergraduate pharmacology degree and had the amazing opportunity to work on a 12-week lab-based project. investigating potential mechanisms of colon cancer development. I was surrounded by such inspiring and driven scientists, all working on different topics and trying to understand the mechanisms and pathways behind how diseases develop and also how to treat them. But they were all working with one common goal in mind, which was finding ways to improve life for others. In December 2017, I began my PhD, also on the topic of intestinal disease. And I've always been drawn towards learning about the gut because of my own personal experiences with gut health and how difficult it can be to diagnose and treat certain conditions. But what can also be difficult is talking about these conditions in the first place. And in fact, many people with troubling digestive problems won't even mention them to their doctor because they're too embarrassed or they don't think it's an important enough thing to talk about. But sometimes, though, these digestive problems can be an indication of something more serious or even life-threatening, such as inflammatory bowel disease or cancer. So it's really important that we take them seriously. But even if a condition isn't life-threatening, it can still have a serious impact on a person's quality of life. Imagine living with a condition where painful, watery diarrhea, cramps, and severe bloating were a regular feature of daily life, where your ability to socialise, work, and travel were all severely impacted by suddenly and urgently needing the toilet, and frequent accidents. Unfortunately, this is the case for many people suffering from intestinal diseases associated with chronic diarrhea, which can leave people feeling isolated and depressed. And I know sometimes when we hear the word diarrhea, our instinct might be to laugh or to maybe feel uncomfortable. But this is something that 5% of the population are believed to suffer with at any given time. So it's really important that we raise awareness of these conditions so that more research can be done and more effective treatments can be found. The gut is the largest immune organ in the body, with a surface area that's equivalent to that of a small studio apartment. But the gut immune system is key to maintaining a healthy gut, but it's also critical for your overall health. Both the small and the large intestine are lined by a single layer of cells called the intestinal epithelium. It's like your skin, but it's on the inside, and it protects your body from harmful toxins that may be in the intestinal contents. It's also really important for regulating the transport of water in and out of the body, and diarrhea happens when this process becomes dysregulated. My PhD focuses on a protein found in the gut called the farnesoid X receptor, or FXR, which is known to control how the intestinal epithelium functions. It protects this inner skin, and when activated, it's been shown to be effective in preventing diarrhea. So the main aim of my project is to find out how this works. But what if there was also a way to increase the amount of FXR in our cells, and even better, a way to do this with food? This could allow for further activation of the FXR and even more health benefits. In our lab, we've been investigating a group of dietary phytochemicals, which are compounds found in plants and food that are believed to have beneficial health effects. And what's really exciting is that one of these compounds was found to increase the amount of FXR in some of our experimental models, and it was also found to further block the mechanisms that cause diarrhea. We know that eating a good diet is essential for gut health, but our aim is to be able to further harness these important phytochemicals for development as targeted nutraceuticals for the treatment and prevention of intestinal disease. Throughout my time here in RCSI, I've had the opportunity to work with some really amazing people while doing this incredible research that is challenging, but extremely rewarding. What I love most about my research is knowing that I have the ability to make a difference to people who are struggling with intestinal disease. By sharing the work that I do, I hope to shine a spotlight on these intestinal conditions so that more can be done to bring about effective therapies, and hopefully make people feel less afraid to tell their stories. Thanks for listening.
Thanks, Jessica. That was brilliant. So now, I suppose we just thank all our presenters in this session for such interesting and thought-provoking discussions and sharing their research stories with us. So we're now going to take some questions that have been submitted to the Q&A box. And just remember, if you'd like to ask our speakers anything about their career journeys or indeed their research, please submit your questions. So maybe if I go to our first speaker, so Lyndsey. Lyndsey, there's a question about the involvement of patients, as you mentioned, patient samples in your research. So are patients happy to be involved in your research? And do you think this is important for both researchers and patients?
Yeah, that's a brilliant question. I think that, patients, they're really happy to be involved. They get a real sense of involvement and interest by being actively, taking part in the research. And then on my end, I get the patient's perspective from their involvement. And this is really great. And it'll kind of go more so towards personalized medicine. And I think, it's a brilliant way forward to have, patients more involved in all of this research. I think it's beneficial to both parties.
Great, I agree entirely. Okay, so if we move on, we have a question for our second speaker, so Camille. So there's a lot of interest about the use of artificial technology in, I suppose, research and medicine. But is there a potential that artificial technologies could take over and that we may not need or rely on the scientists and also the pathologists as much anymore? Camille, can you hear us?
Yeah,
Oh, we could hear you, Camille, and then you went muted, I think. I think there might be some technical issues, so maybe we will just move to a question for our third speaker.
I'm here again. Sorry if you want me to answer. I think it won't turn off again.
Yeah, we're good.
It's actually a really common topic among pathologists and in the digital pathology world. But the consensus really is that, these artificial intelligence algorithms, they need to be trained by research scientists and pathologists. And this takes a lot of work for their training. So they can't train themselves. That's one thing. And another thing is it's really that they're going to be aiding the pathologists and aiding the research scientists. So the human is still the boss and the AI is the helper.
Great, so we're not all out of a job just yet.
No, we're not out of a job.
Thanks, Camille. Okay, and our final speaker, Jessica, we have a question for you. So if you're still with us. So there's some interest that with the rise of fake news and fad food trends, we're wondering if the general public are more aware of the beneficial and bad foods for health or whether we're more confused than ever.
Yeah, I think that's a really, really good question. Because we're hearing new information every day. Oh, you shouldn't eat this, you shouldn't eat that, like bacon causes cancer. But I think for the most part, it's important to just keep things simple, limit the foods that we're told are bad for our health, or just eat them in smaller quantities, but you don't have to cut them out completely. And maybe focus more, like what we'd be interested in are the likes of vegetables and fruits, beans, things like that, more plant-based options. But I think it can be quite confusing for people out there to make the distinction between good and bad foods. So just remember that there's no such thing as a bad food. There's just, you can just have too much of it.
Great. So everything in moderation.
Yeah.
Thanks very much, Jessica. So with time in mind, I think I will now close out this part of our Discover RCSI Research Stories event, and I will hand over to our next session, which is focused on health and societal challenges and will be chaired by Dr. Judith Coppinger. So please join us for this.
Hello. So I'm Judith Coppinger and I will be chairing this final session, Health and Societal Challenges. And our first speaker is Deirdre Fitzgerald-Hughes, who's a senior lecturer in the Department of Clinical Microbiology. And she's going to give a talk on superbugs, tiny creatures, big problems.
You're currently muted there, Deirdre.
Okay, now can you hear me?
Yes, thank you.
Brilliant. And I don't know if you can see that slide there, can you?
Yes, indeed, thank you.
Okay, so sorry, yeah, I'm Deirdre, and I worry about antibiotic resistance. And so in my research group, we discover new ways of killing bacteria and better ways of treating or controlling superbugs in our hospitals. So what about antibiotics? Well, antibiotics are the medicines or the weapons that we use to kill bacteria when they cause infections. And antibiotics are amazing. So my little boy was in hospital with pneumonia some years ago, and he got antibiotics to help him clear that infection, and he really bounced back. So antibiotics are also used to prevent infections, and that's again really important. So when I had my C-section to deliver my twin boys, I got antibiotics to stop me from getting an infection. And I can't say I bounced back, but I was okay eventually. But you know, there's lots of other examples of operations that take place. So think about getting your appendix out, or getting a pacemaker fitted, or all of the knee and hip operations that we take for granted, and even organ transplants. So all of these operations are life-saving and life-transforming, and they can be done safely because of antibiotics. So they really are very precious medicines, and I want it to stay that way. So there is a downside to antibiotics, and it is that bacteria don't want to be killed. So bacteria will do whatever they can to avoid being killed when they're being attacked by antibiotics. And the ones that we call superbugs, they've taken this to the next level. So they have developed really sophisticated ways of avoiding being killed with antibiotics. So for example, some of them are able to directly destroy the antibiotic structurally, and others are able to activate kind of shields or defenses that will help them to avoid being killed. And so bacteria have developed these skills over, or what I like to call superpowers, over generations and generations of being attacked by the antibiotics that we use. And when you think about it, so hospitals and especially intensive care units where the sickest of patients are treated, that's where antibiotics are used the most, because of course that's where they're needed the most. But if you're a bacteria, there's a very high chance, of course, that you're going to be killed in a hospital and in an ICU. But if you survive, there is a chance, or there's an opportunity for you to develop those skills that I talked about, and to really kind of test and perfect those superpowers to become a superbug. And that's where the real challenge for doctors lies. So really treating infections is hard enough, but treating infections with superbugs is much more complex. And I suppose simply put, we're really running very low on antibiotic choices to treat superbugs. So what are we doing in my research in the fight against superbugs? Well, one thing we're doing is surveillance. And what that means is that we are hunting them down. So we are going to the hospitals and we're finding out in the wards, we're finding out where they hide, and we're finding out how they manage to hide from us. And so that involves taking lots of samples from the ward, close to patients, in the ICU as well. And we take all sorts of samples. We even go down drains, and we take samples from the patients themselves. And when we come back to the lab, we identify those samples that contain superbugs, and we study them. And when we do this in a very systematic way, we're able to gather evidence, and we're able to present that evidence to the decision makers. So, for example, to hospital management, or even through our networks to government agencies. And that really helps them to make recommendations for improvements based on the evidence. And so the other really exciting thing that we're doing is we are discovering new ways to kill superbugs, ways that override these kind of superpowers that they have. And the beauty of that is that there's a real element of surprise for the bacteria because they don't see it coming. They haven't seen these new structures before, and so they're not able to, as yet, develop those superpowers against them. So that's really exciting and gives me great hope that we'll continue to be able to treat infections even if they involve superbugs. And finally, to say that this is a real team effort. So our researchers and myself, we work with the doctors and nurses and all of the other health professions that look after patients in hospitals, including those that work behind the scenes, like the medical scientists. And we also work with other scientists. So I'm a microbiologist, but we also work with chemists and with engineers. And that's when the real magic happens, when you're getting all those multiple perspectives focused on this really important problem. And finally, just to say that most importantly, we work with patients because, of course, they're the real experts. They've had infections, they've been in hospital. So not only do patients help us as participants in our research, but they also advise and provide their expertise, and that really helps us to make sure that we're asking the right questions and that patients are really shaping the research that we do. So thank you very much, and I'll be happy to answer questions at the end.
Judith, you were muted.
Apologies. Sorry, on to the next speaker, Michelle O'Toole. She's a researcher at the RCSI Sim Centre for Simulation, Education and Research. And her talk is Stress, Not Always a Bad Thing.
Hi everyone, just bear with me while I bring up these elusive slides. Hopefully that's ready to share. Oh. Okay. So thanks very much. Hopefully that's you can see those slides. Can somebody confirm that you can see that for me?
We can.
Thanks very much. So usually when people think of stress, our minds go to the negative aspects that can potentially cause us to become unwell. And this unfortunately is a reality. But our body's stress response can actually help us to perform and work in our favour. And this is especially relevant in the emergency services. For the last 12 years, I was a firefighter and paramedic before making a leap into full-time research with RCSI this year. And everyone knows that emergency services work can be extremely stressful. But as a fresh-faced recruit, I learned this the hard way. My very first call out on my very first day as a recruit was a road traffic accident where a young man sadly lost his life. And a few things from that day have stayed with me and directly influenced the course of my research ever since. So I'm going to share some of those reflections briefly with you today. So on that very first call out, I remember jumping out of the fire engine, full of enthusiasm, yet finding myself frozen to the spot when I saw the incident in front of me. I could feel my heart beating faster in my chest. My breathing got harder, sweat was dripping down my back, and my eyes opened wide, taking in my entire surroundings. Adrenaline was pumping through my body, but instead of responding like I was expected to, I froze until one of my colleagues took me under his wing and showed me the way. I now know that this is a perfectly normal reaction to a completely abnormal event. I also remember feeling an overwhelming sense of sadness and guilt when I realised that this young man would die. Emotionally, I felt very unprepared for this type of event, and I received some fantastic peer support from my colleagues you'll see here in the middle picture. They both listened to my concerns and shared stories about their first stressful incidents. My personal reactions to these events sparked my interest in learning more about post-traumatic stress, or critical incident stress as we sometimes call it in the emergency services world. But when I started studying and researching, I kept coming across the bad news, and there was a lot of bad news. Firefighters and paramedics are 3 times more likely to experience serious mental health issues than the general public. Emergency services personnel are more likely to abuse alcohol and other substances, and further research suggests that firefighters, paramedics and police officers are more likely to die by suicide than the general population. So I started my research in these areas with my colleagues and found that most of those claims were unfortunately true in the Irish context. What I hadn't expected to find was that there could actually be a positive element to all of this trauma. I noticed that experiencing all these incidents changed my perspective. And as corny as it sounds, it gave me a much greater appreciation of life and the wonderful relationships that I'm fortunate to have. I hugged my kids tighter after every shift, and I learned not to take even the simplest things for granted. Social support is proven to be one of the most important factors in mitigating stress in all walks of life, not just for first responders. And we heard a lot about this during the pandemic. Hopefully you've all realised the benefits that keeping in touch with our social connections can really help to maintain our mental health. Having loved ones, whether that's family, friends or colleagues to talk to and to let off some steam with, can actually protect us from the negative effects of stress. Professor Stephen Joseph in the University of Nottingham introduced me to the concept of the shattered vase, which you can see in the image on the right-hand side of the screen. And this is a lovely analogy of our options following a stressful event. So the idea is that you have this beautiful vase, or you might say vase, depending on where you're from, and one day you accidentally knock it off a shelf. It smashes into smithereens and you're absolutely devastated. But you have a couple of choices. Number one, do you pick up the vase and try to glue it back together, knowing that water might seep through it and it'll never be the same again? Two, do you discard the vase, throw it in the bin and try to forget that it was so precious in the first place? Or three, do you pick it up and transform it into something new, like a mosaic on the wall that you can still admire and you can still value? This third option describes post-traumatic growth, the positive things that can come from stress if in time we can open our minds to the possibility. So we don't just recover from the stressful event and go back to how we were before. We can actually surpass that pre-trauma state and grow from the experience. So to summarise my research journey to date, I've studied aspects of both post-traumatic stress and post-traumatic growth in my firefighter paramedic colleagues. I've also interviewed their peer support workers, those colleagues who provide a listening ear following the stressful incidents. And we now know that first responders lean on their spouses, their family members, their loved ones, their close friends for support. See my wonderfully supportive family here on the right-hand side of the picture. So in conjunction with the Movember Foundation and RCSI Simulation, our next step is to try to find the best ways to care for these carers, to increase the social support of both family members and first responders, and hopefully help them all to grow too. Many thanks for your attention. I'd like to take your questions at the end.
Thank you. Thank you very much, Michelle. That was a really interesting talk. I really like the vase analogy. Okay, so on to the next speaker. It's Eunice Phillip, and she's a research assistant at the Department of Epidemiology and Public Health, and she's going to name her talk as Chasing the Voice.
Thank you all. I'm going to try and share the screen now. Okay, so good afternoon, everyone, and thank you so much for the previous speaker. It's been amazing listening to the amazing work that you guys have been doing. So for me, when I talk about chasing the voice, I remember how the dream started. And it wasn't a big bang moment like I've been hearing all along. It actually came when I was about 4 or 5. And I remember having pneumonia, and there was this amazing doctor that came in and actually spoke to me. And it took me decades later to actually get the hang of what that meant. To me, it was just this good-looking doctor that actually paid attention to me and asked me if I was feeling okay. And I knew then that I wanted to be a medical doctor. I had to give back something. And this dream, this passion, and this desire took me through 3 continents, across 3 different college degrees, and over 3 decades. And it was in the 10th year of my nursing degree that it actually started to make a little bit more sense. And that sense came from a tuberculosis patient who would come into the clinic every 2 weeks, never missed an appointment. And at the end of 6 months, she still wasn't feeling any better. And you think something is not right here. And then there was the HIV patient that all through the year, he's been consistent with his medication. He's been exercising. He's been eating well. He wanted to be better. And then all of a sudden, he dropped out of the program. And then there was this mom of two who was whisked the two children into the emergency department almost every Friday night. There is always something wrong. There is cough, there is respiratory disease. And you're wondering what is going on Friday nights in these people's home. But having been conditioned to treat and let go, I missed those voices. There was something that needed to be heard that was not heard. And that stood with me. And then I decided that it's time to go and figure out what this was. And that's how my research journey started. I came over to UCC and did my master's in public health. And lo and behold, I got a professor that was interested in household air pollution. And I was like, wow, maybe that was why that woman was not, the children were not feeling better. I never asked her the type of fuel or what they used to cook in their home. I just treated the kids and they went back home. I never called that TB patient and found out what happened. Like, you take your drugs, something is missing. Or that HIV patient, I never connected. Something went wrong there. I missed those voices. That is my chase. My current study now is now giving me a better platform to actually be able to explore these voices of the people. I'm working on a project called the Smokeless Village, and the project is very interesting. We're looking at household air pollution and ambient air pollution, which is the outside quality of air. In a small village in Malawi, this village has no electricity, no potable water. There is no LPG, so you can't turn on the gas and something is happening on your cook stove. And when I got the opportunity to work on this project, I said, I have found my needle in the haystack. Things are beginning to make sense. The 30 years or decades of running over something that I wasn't understanding was meant for this purpose. And when I now got the opportunity within this project to actually look at a PhD option, it was a no-brainer. I wanted to do something about the voice. So I picked up community-based participatory research. And what this approach is trying to bridge the gap between researchers and the community that we are researching, or the people that we are researching. In Europe, they call it PPI, the patient public involvement, trying to get the voice of the people. And it's so funny, like sometimes when I sit down and I think back and I'm like, wow, things actually fit together. In the next few months, I'm going to be going to Malawi to collect data. I am excited, I am anxious, I am terrified, but there is this core that I've carried with me all these years, all through medical school, that didn't work, all through nursing school, that these people's voice is actually that gap that we can feel. It's not about the medication, it's not about the intervention. It's about what they want to say. And that is my goal with this research, that I'm able to capture the voice of these people and integrate that in the way that we plan our research, in the way that we conduct the research, in the way that we present the outcome of our research, both to the community and also to the policymakers. Not as a tick-box process, not at all, but as a genuine collaboration between researchers. I want to learn from these people. There is a reason why that woman just doesn't think that she needed to take the child away from the fire. Maybe there is no extra room. I didn't explore that. Research is giving me the opportunity to go back and make amends for that. And there is nothing more that I would like than for you guys to follow me on this journey. I'm just 1 year into the PhD and it's so exciting already. I'm hoping you guys can follow me on this journey and travel this, and hopefully we can capture the voices of these people and integrate it into the way that we do our research. That is my spin. I didn't get to be a medical doctor, but what research is giving me is the opportunity to be a doctor for the voices of the people that I left behind. So thank you for listening to me, and I'm open to all your questions. Thanks.
Thank you. Thank you very much, Eunice. That was a lovely talk, and you can get some questions later. On to the final speaker of the panel. It's Ciara Fallon, a postdoctoral researcher in the Department of Molecular Medicine, and her talk is Gut Feelings, Cracking the Code to More Natural Medicines.
So I hope everyone can see my slides there. Good afternoon, everyone, and thank you for tuning in to hear my research story to date. My name is Camille Hurley, and I've recently just completed my PhD in the Royal College of Surgeons. But how did I end up here, and what sort of research have I been involved in? Well, I have been interested in science since a young age, and I have experienced firsthand the amazing impact science and technology can have on people's lives. When I was 5 years old, my family realised I was having difficulty hearing certain sounds, and afterwards went on to discover I was profoundly deaf. As the years progressed, my hearing deteriorated, and when I was 16 years old, my deafness was severely impacting me, and my hearing aids were no longer useful. This was when I was referred to the Cochlear Implant team in Beaumont Hospital, who after various assessments decided I met the criteria to receive a cochlear implant. So a cochlear implant is a device which provides access to sounds for people who suffer from hearing loss. There are two parts to a cochlear implant: the inner surgically implanted device, which when coupled to an external processor, provides access to the speech, noises and sounds of everyday life. So as you can see here on Mr. Koala, he wears his external processor on his ear, and then he has a device called a coil, which attaches to the side of his head via a magnet. So the processor collects the sounds, and then the sounds are transmitted into an electrical message, which is passed through a wire called an electrode directly into the auditory nerve. The auditory nerve then sends a signal to the brain, and the brain interprets this electrical signal as sound. While the very first cochlear implant was invented in 1957, the design which formed the basis to the modern cochlear implant used in Ireland today came about over 20 years later in 1978. This goes to show that research into science and technology is progressive, and when someone discovers something, they put down a building block which future scientists can further develop, and all this research can lead to amazing life-changing results. This idea of making a contribution to a research area which could help progress the research to ultimately help patients is what drives me as a scientist. Now my research is also still about communication, but through a different manner. My personal area of research is in intestinal diseases and the gut. So why is the gut interesting? Well, we know the gut is full of different microorganisms, and these microorganisms include bacteria, viruses, and fungi. In fact, there are more bacteria cells than human cells in your body, and each of these bacteria can play different roles, and there are over 1,000 different types of bacteria in the human gut. These bacteria in your gut are alive, and they talk to you. Most of these gut microorganisms are found in a particular pocket of your gut called the gut microbiota. In recent years, research into the gut microbiota has shown that it may play a role in controlling many aspects of your body, such as how you fight infection, your brain health, and also how you digest food. Also, research shows that food can actually impact your gut microbiota. So your diet can actually impact the diversity and the amount of different classes of bacteria, fungi, and viruses in your gut. When you eat healthy foods such as fibers and plants, you can enhance the good bacteria in your gut. And when you eat unhealthy and fatty foods, this can enhance the bad bacteria in your gut and lead to bad health. So these bugs in your gut modify food to send signals to their human. And in states of intestinal disease, your gut microbiota can display unhealthy and harmful properties. So my research area is looking into the idea that foods or parts of foods could be used to treat or prevent intestinal disease, and along with this, revert a harmful gut microbiota back to a normal and healthy state. We would like to do this through the use of nutraceuticals. So nutraceuticals are foods or parts of foods which can be used as medicines to treat or prevent disease. You may have never heard of the word nutraceutical before, but actually, if you drink fortified vitamin D milk or take any food for your health, like jelly vitamins, these are nutraceuticals. So far, my research has shown that a common food crop, which we all probably eat in our diet, could have the potential to treat intestinal disease. Our future research is focusing on how are these gut bugs in your gut microbiota talking to you, and how are they sending signals? And can we decode these signals that the bugs send to humans after consuming different types of food? When we understand the language that bugs and humans communicate by, we will be much better able to understand disease and how to fix this. So just to finish off, I'd like to mention I'm incredibly grateful to be a part of such an amazing scientific community here at RCSI. And looking at the accomplishments of the amazing women who have come before me helped me to remember to never give up on my research when things go wrong and to keep on trying. Thank you.
Thank you very much. That was a great talk. And so now what we're going to do is we're going to have a few different questions for everyone. The first question is I'm going to address to Deirdre, and that is what can ordinary people do to make sure they don't get superbug problems?
So that's a great question, and I'm glad to answer that. So I think what it is really important, first of all, to say that you should always follow the advice of your GP and your doctor. But what I would also say is don't always be thinking that you need antibiotics. And what I would say is, unlike what Donald Trump thinks, antibiotics do nothing for viruses. So a lot of the infections that we get, you know, the kind of colds and flus, antibiotics won't do anything for them. And really what you're doing there is just giving those, you know, bacteria a chance to develop those superpowers that I mentioned. And also, if you are taking antibiotics on the advice of your doctor, just make sure to finish the course. There should be no situation where you know, there's some left or that you have some to share with somebody else. That just shouldn't happen. But I would urge people to have a little root around their drawers at home and see if there are any antibiotics, and I think you'd be surprised. And if there are, just bring them back to your chemist, and they'll get rid of them in the right way.
Thank you very much, Deirdre. So now I have a question for Michelle, and just sort of a general question was just do you have any recommendations for people if they find themselves in a very stressful situation?
Absolutely. I think the best answer for that, Judith, and it's a really common thing that people are finding especially over the last 18 months, is to try and talk it out. Talk to somebody. It doesn't have to be somebody professional. If you're more comfortable speaking to a family member, speaking to somebody that you trust, that will help you to get things off your chest. And then if needs be, other people can potentially help you get the help that you need if you do need further mental health and professional help, for example.
Thanks very much, Michelle. And then Eunice, there was actually two questions for you. So the first one was, from your experience, to what extent do you think searching for the patient's voice will influence their health and well-being?
To me, I think it's the antibiotics needed for the bacterial infection. You know, I mentioned that I didn't realize what that doctor did for me until much later. He could have asked my parents what's wrong with her. But there was something about it that I look back now and I realized that he actually gave me a sense of being. And when you feel validated, we all know all these are psychology, you know, capacity building, you know, human endurance, all comes from this internal validation. When somebody asks you a question, what do you think? There is something powerful about that just makes whatever it is that's going to come after that much more easier to deal with. If I asked that woman that day and said, oh, I noticed every Friday you bring that child in. Is there something going on Friday? Oh, we always have a party where we all cook around the fire. You know, one, I'm validating who she was by doing that. And at the same time, I am getting data to be able to actually address that. So if we hear those voices, then we can plan better as researchers, as clinicians, as whatever it is that we do. Without that voice, we are just adding volume of papers on research papers on causes of diseases. That's the way I think anyway. That's my disclaimer.
Thank you very much. And there was just one other quick one. It's probably not a question you can answer very quickly, but just really it was there anything particularly striking you learned from listening to the community?
you learn who you use, your own faults or your own limitations, because they bring this wealth of knowledge, you know, to you as a researcher that you would never found in a research paper. That's the best thing that I've learned, yeah, from the community.
Okay, thanks. Thank you very much, Eunice. And I had a question for Kira. I typed it into the chat box, and it was just, Kira, do you think natural products such as probiotics will help the gut? In, for example, yogurts and so forth?
Kira, you're muted.
I think she's muted.
Sorry, I muted myself so I wasn't whistling around and everyone else dancing. Yes, I do think that probiotics, such as natural products and probiotics, can help the gut. But they could help enhance the good, healthy bacteria in your gut. Yeah. And I do think that they can make a difference. Yeah.
Thank you very much. And thank you to all the speakers for really great talks in this session. And I think this actually wraps it all up. Lucia, or does anyone else have any further comments?
Yeah, I'm happy to close us out. So as Judith said, this brings us to the end of our Discover RCSI Research Stories event. So just on behalf of both myself, my co-chairs, Louise Keating and Judith Coppinger, we'd like to thank all of our speakers for taking the time to share their research stories, but also their personal career journeys with us today. And also to all members of the RCSI Women's Network, and especially the Equality, Diversion and Inclusion Unit at RCSI for their help in us bringing this event together. And thanks to all of our online attendees for listening today and engaging, and we hope that there was something for everyone in today's session. Thank you very much.